This next section talks about prokaryotic cell structure. So all organisms, at least all living organisms, are composed of cells, whether it's one cell or multiple cells. A cell is the smallest unit of life that can function independently. There's different types of cells that exist, and one of the major kind of breaking points for what defines a cell is whether it's eukaryotic or prokaryotic. A eukaryote or a eukaryotic cell is a cell that has a nucleus and membrane-bound organelles. So it has a nucleus and membrane-bound organelles. While a prokaryotic cell or a prokaryote is an organism whose cells do not have a nucleus or membrane-bound organelles. We'll learn about what the membrane-bound organelles are as we go through a couple more slides here, but eukaryotes are going to be our cells, plants, algae, protozoa, fungi, while prokaryotes are really just bacteria and archaea. Now, viruses and prions are considered acellular. When you put the letter A in front of something, think it's saying like the absence of. So acellular is the absence of a cell. These are not cells. Viruses are just small infectious particles. All they are a nucleic acid that's surrounded by a protein coat. So a virus is an infectious particle. It's a nucleic acid surrounded by a protein coat, and it is acellular. It's not a cell. A prion is a small infectious protein. You may not have heard of the term prion before, but I'm sure you've heard of the disease, mad cow disease. So mad cow disease is a prion disease, or chronic wasting disease is another one. Scrapie in sheep, that's another one. And there are human forms of this, like Creutzfeldt-Jakob's disease and familial insomnia. But these infectious proteins are self-replicating, so they're not even an organism, and the protein, it's just a protein that's making copies of itself continuously. So prions are pretty interesting, and there's not a lot of research out there on them because they don't really kill people that often. It's such a rare disease that there's not a lot of people that want to put up money to fund research in that field. But prions are infectious proteins. So starting with basic cell structure, the most basic organisms are the prokaryotes. So prokaryotes are extremely small. Again, if you remember, I said that they are archaea and bacteria. And we typically classify these prokaryotic microorganisms based on their cell shape. So we have coccus, which is spherical, bacillus, which is rod-shaped, spirillum is a spiral shape, and if it's rigid, if it's like not flexible, then it's considered spirillum. But a spirochete is a flexible spiral-shaped bacterium or prokaryote. And then we have this cell envelope, and the cell envelope has multiple layers. This is actually from the inside out. So this is the innermost layer, and this is the outermost. So the plasma membrane, or the cell membrane, is something that we're going to talk about in this PowerPoint slide. So we'll learn all about its structure. Talked a little bit about phospholipids in last week's lecture, so those phospholipids make up the plasma membrane. Then prokaryotes have a cell wall. So a cell wall really defines the shape of the cell, so that gives it its most structural shape and support, so that's going to give it its cell shape. And then we have the glycocalyx. The glycocalyx is an external matrix of proteins, carbohydrates, lipids that make up the outermost surface of a prokaryotic cell. Now, eukaryotic cells also have a glycocalyx, but we often don't talk about a glycocalyx when we're teaching classes like human anatomy and physiology. But any receptor proteins that stick out, like if you have hormone receptors or receptors for neurotransmitters or anything like that. So those are all considered part of the glycolytic. So it's kind of this external layer and this association of a bunch of different molecules that are just found on the surface of a cell. In the case of bacteria, the glycolytic plays a major role in allowing that bacterium to stick to you in order to cause an infection. Because if this microorganism can't stick to your tissues, it's just going to be flushed out of the body. So the glycolytic has a predominant role in allowing that bacterium to stick to you. Also, it helps it hide from your immune system. Sometimes the glycolytic consists of this gel-like layer called a capsule, and your immune system can't recognize it. It doesn't really know what it's dealing with when it sees a capsule. So that's kind of a sneaky way that bacteria can get inside of you and stay inside of you. Now this is a picture of the different structures of prokaryotes. So this is a coccus. If you want to say there's multiple ones, you would call them cocci. Bacillus, that's one of them. If there's multiple, you would call them bacilli. And then we have spirillum and spirochete. So this is a picture of some of the structures that make up a prokaryotic cell. We have ribosomes that are the site of protein synthesis. Fimbria are hair-like projections that allow for these things to stick to your surfaces. The nucleoid is the region where you would find the genetic material. Then you have the plasma membrane, the cell wall, the glycolytic, which in this case is composed of a capsule. Some also have a flagellum, which is a tail-like structure that allows for this microorganism to swim or allows for motility. So inside of the cytoplasm, we're just going to define some of these terms here. The nucleoid. The nucleoid is the region where you have DNA. So it's the region where you have DNA. If you think about a prokaryotic cell as a studio apartment, if you've ever been in a studio apartment, it's just one big room, and then you may have like a little tiny separate area for the bathroom, but basically everything's open in one area. When I first moved down to Texas, I lived in a studio apartment. So basically the kitchen was open to the living room, which in the corner I put my bed, and it's just a tiny box that you live in. But that kind of area, if you can think about that and visualize that in a studio apartment, that's kind of what the inside of a prokaryotic cell looks like. Now, I put my bed in one corner of that studio apartment. Well, if we call that the nucleoid, that's where the bacterial cell would put all of its DNA. It's not enclosed by walls. I didn't have a separate bedroom in that apartment. It's just a region. It's just an area where we happen to pile all of the DNA. It's just the part of the box where I put my bed. So that's the nucleoid. It's just a region where you have the genetic material. Plasmids. Plasmids are small circular pieces of DNA that are non-essential, but they provide extra genes to the microorganisms. Plasmids are actually pretty cool. So if I'm a bacterium and I develop this resistance to an antibiotic, in my mind, as a bacterium, I'm thinking, okay, safety in numbers, safety in numbers. So if I happen to give this antibacterial or antibiotic resistance gene to all of my friends, then I have a lot of other microorganisms around me and I would be safe. It's kind of like, let's say you were resistant to bear attacks and there was bears everywhere. Well, you know, you're more likely to get attacked if it's just you all by yourself. But if you had a whole bunch of friends around you, you only have to outrun your friends. You don't have to outrun the bear. So safety in numbers. What these plasmids are capable of doing is if I have a plasmid and I have a gene on that plasmid that's resistant to an antibiotic, I can start handing that out to all my friends. So now I've got tons of other bacterial cells around me and I'm safer. And additionally, they just provide like extra genes. We use them a lot for genetic engineering. One of my major jobs up in UW-Madison was isolating plasmids, inserting new genes into those plasmids, and then inserting those plasmids into new bacterial cells. It's called genetic transformation. So they're kind of cool, and they have a lot of uses, but predominantly out in nature, they're to transfer genes between bacterial cells so that we can provide some type of benefit or survival benefit, so that now you have more of that bacterial cell around and there's safety in numbers. Ribosomes are responsible for protein production. Ribosomes usually come in two subunits, a small one and a large one. You don't necessarily need to know the size of each one of the subunits for this class, but basically they're going to squish together to make a protein. We're going to learn all about that process later on in the semester. External structures of bacterial cells, we have the flagella, which is a tail, fimbria or fimbriae, which are hair-like projections that allow the bacterium to stick to your surfaces, or to stick to surfaces of anything, really. And then we have conjugation pili. Conjugation pili allow for the transfer of genetic material from one bacterial cell to the next. Kind of a funny way to think about this, but conjugation pili, think of it's a tiny bacterial penis. So what does a penis do? It is going to inject genetic material into the vagina so that conception can happen. Something similar happens with a pilus. So although bacteria don't mate, like they don't have babies, and there's not boy bacteria and girl bacteria that come together and exchange this genetic information, you have one bacterial cell that will grow a pilus and insert it into a neighboring bacterial cell, and then it will inject that piece of DNA or genetic material into the neighboring cell. This is how plasmids can be transferred from one bacterial cell to the next. And as soon as you do that transfer, now that other bacterial cell has an added benefit. So it just got some type of extra genes that are usually beneficial to the microorganism. So conjugation pili aid in the transfer of genetic material from one bacterial cell to the next.